you for the introduction. I would like to thank Miguel for the opportunity to be here for this presentation. Actually, the title is a bit general. I would like just talk about two topics, two research areas in which I'm working on, actually. The first one is using classical potentials, classical simulations, and the second one is a quantum approach for tackle a novel system that I will talk about later. So let's start with the first one, which is just how to design some kind of biopolymers similar to natural coating polymer. So in material science, actually scientists are all the time inventing new sophisticated materials, and recently the biomimetics is a general term that have been used just to learn from the nature. We have many things that we have many solutions that we can learn from the nature. Actually, polyesters are very interesting system because they are because of the biodegradability, biocompatibility, and non-toxicity. So, and for that reason have been used in different applications. We are interested in protective biomaterials. In particular, if we look at most of the vegetables or fruits in the nature, we find that they are well protected from different impact, chemical, physical impact. Let's see a tomato. In a tomato, we see the skin. There is a layer, relatively thick, that protect from this kind of attack. This skin is called basically or formed by the cuticle. The cuticle is this extracellular layer that protects the fruit and the leaves. The biopolymer coating is the main chemical structure that form this system. So the coating is a biopolymer that is a high molecular weight polyester formed by different hydroxyalkanoic acid. And the interesting thing for this system is that coating is amorphous when we look at the fruit or the leaf. But initially, at the early stages, it formed a lamellar structure. This is the key point for my talk. Let's go back to the monomers for forming this biopolymer. From the depolymerization, we can find this kind of molecules, which are the main monomers. All of them have carboxylic group, terminal carboxylic group, and one hydroxy group that in a terminal position or in a lateral position, different kind of possibilities. How these monomers can form the polymer? Well, they use the self-assembly process, which is a well-known recurrent mechanism in nature to build complex structure. We have used this strategy as well as a bottom-up technique like in electrochemistry to build specific monoligands with different properties. But how does it work in the nature for this kind of biopolymer? Well, just reminding the monomers that we have considered in this study, including one of them which is palmitic, doesn't have a hydroxy group, and all the others have a specific hydroxy group in strategic positions. We have examined the position of this hydroxy group to understand which is the role in the process. And as I said initially, the important point is that the biopolymer is formed at the early stages in a laminar way. That means that initially the system used like a support, a substrate, to start to the condensation process, the esterification process. So we have used in the lab this strategy using a substrate which is the moscovite mica, which is why we use this. Basically because it's cheap. We are trying to get a way to fabricate a biopolymer in an industry scale. So basically, initially we used to try to mimic the laboratory strategy using cheap materials. The moscovite mica also has a particularity, which is that the binding between the monomers and the substrate are very they are not very intense. They are very weak bond, So the interaction will control basically the process are the lateral interaction between the monomers, and not the specific or covalent interaction like in the thiol cases for thiol molecules on gold, for example. We have studied this assembly process using experimental techniques and also atomistic simulations. So I'll show some of the results. Using the most simple monomer, which doesn't have the hydroxy group, the palmitic acid, shows at when we look at the AFM images, found different dendritic islands, which has a very low height, basically around 15 angstroms, or even smaller. And the simulation shows us that there is no effective bonding between the carboxylic group at on the surface. We can mimic in some way how these dendritic islands are formed. This is because this hydrogen bonding is not very effective. The carboxylic group disrupts a compact or homogeneous network. When we add a hydroxy group at the end, a terminal group, we get this molecule, which is also a monomer in the natural coating. We find now that another effect. We find that also multilayer structures are formed, and we find that the steps, the height of the steps are around 20 angstrom. Again, simulation helps to understand how these peaks, which correspond to monolayers, which because of the height, the molecule form very uniform structures, differently from the palmitic acid. And also the multilayer structure can be seen from the simulation that are not formed for the palmitic acid. And they are very effective for this second molecule with the hydroxy group. The hydroxy group in the terminal position interact effectively with the carboxylic group coming from the second layer, forming these nice structures. What happens if we add another hydroxy group to the system? Basically, the nature is clever, and because of what we can expect, this is the main monomer extracted from the depolymerization. What we'll expect? We expect that the coverage of the system of the marker is very effective. It's very effective. So the first monolayer develops very fast, covering all the surface. And also the multilayer system is also observed in this system. So again, the simulation can show us that in the monolayer system we find a very uniform networking, a hydrogen bond network from the lateral hydroxy groups, which is in yellow, and on the top is a second hydrogen bond network forming by the terminal hydroxy groups. This is a design of these interactions schematically, and this is what explains the effectiveness of this molecule to form the biopolymer. We have also two experimental interesting observations. The first one is from the XPS, which we observe a reduction of this cyanide, which means that we are losing oxygen. What does it mean that we are losing oxygen? Basically, that we are losing water. Why? Before the answer, we have another observation. The other observation is from infrared spectrum that looking back at the monomers, if we take an spectrum from carboxylic groups, we would expect this spectrum. However, what we find here is this other. This other doesn't have anymore the cyanide from the carboxylic group, and there are two new spectral fingerprints corresponding to the formation of ester. How an ester is formed? Just remembering from basic chemistry, in the esterification reaction, there is a nucleophilic attack from the oxygen in the alcohol group to the carbon from the carboxylic group, and also the interaction between this hydrogen and this oxygen, forming a water molecule and the corresponding ester bond. To do this in an homogeneous solution, we need a strong catalyst. We need a strong acid, which has the role of a catalyst. But in nature, we don't have this. The fruit and the leaf form the polyester in mild conditions. How does it work? The simulation can help to understand the process. Why? We are using classical potentials. So we are not expecting to observe any chemical reaction. Maybe the reactive force fields that Douglas mentioned in the previous talk would give some different results. But the interesting thing here is not the reaction itself. It is the approximation of these potentials to mimic the arrangement between the two layers. Let's consider a simple system. One layer absorbed to the mica surface, and the second layer, which is developed from this interaction. We can observe that there is a very effective approaching between the different groups that I mentioned. The oxygen from the alcohol group to the carbon to the carboxylic groups. And this can be visualized in the radial distribution functions. We can see how the how the distance which could evolve to the water formation evolves along the time to give a peak. And also this shoulder would correspond to the nucleophilic attack. What it means is that the arrangement, the effective arrangement of the first monolayer helps to the construction or to the building of the system to facilitate the formation of the ester bonds. We also use this strategy to understand another problem in the vegetal biology, which is the origin of the cutting zones. The cutting zones are considered the nanoparticles, vegetal nanoparticles, which are the origin of the cutting. So, initially, as I said, the cutting will evolve in a laminar way, and later on, when the polyester gets in a high proportion, they can start to form this kind of particles, which are called cutting zones. And they are like a nanocluster that can sinter, forming new larger particles. Simulation also show the structure of the interior of these nanoparticles, which is a very esterified liquid-like content, and the shell is a hydrophilic system. Well, all these results help us to suggest artificial monomer to create the biomimetic polymer. We used the aleuritic acid, which has also the carboxylic group, the terminal hydroxy, also the other hydroxy group, and an additional hydroxy group. We expect that this helps to the effectiveness of the system. Why we used this? Because we can buy this polymer, this monomer, for a reasonable price. The previous monomer, the 16 hydroxy carboxylic that I mentioned, it only comes from the polymerization, and it's very difficult to synthesize, and it's very high cost. So this is a very interesting alternative. And the results show that the system works pretty well. They also form multi-layers. And there is an interesting aspect that we can observe from friction atomic force microscopy, in which we observe two different kind of regions. Also, we observed all the other particular structure that I showed initially, multi-layer structure, but I want to put the focus on this. These two regions, low force and high low friction and high friction regions, has an origin that can be understood from the molecular insights. Simulation can show that an older monoligand has this structure, and the determination of this layer is basically hydrophilic. When we have disordered monoligand, which are with height, lower height, obviously, and the behavior is completely different. They have a more hydrophobic behavior. This can be correlated with the two regions. The low friction correspond to the hydrophobic structure, and the high friction to the hydrophilic. This is because of the interaction of the tip with the system. And also, this monomer was used to fabricate the polymer. The polymer works quite well, and we suggested two different routes for the synthetic process, and found this kind of biopolymer, which can be used for different things. Basically, the main one is to cover, for example, metal surfaces, and to contain food. And the chemical comparison between the different properties, chemical and mechanical properties of the natural lutein and the polyaurate are very similar. So recently we also are working on another aspect, which would be to implement coarse grain parameters to model these molecules. This allows us to simulate larger systems. Why larger systems? Because I didn't say, but when we buy the aleuritic acid, if we want to buy it for a cheap price, we buy it contaminated, and many other monomers will be present. From the point of view of the product, it works. When we use this mixture of polymers, the polyester is formed, and we got this plastic. But from the fundamental point of view, the system is more complicated. And then we are trying to study segregation problems that appear when we mixture different monomers. And with atomistic simulation, we are having some difficulties to tackle larger systems. That's why coarse grain simulation could help. Well, this is how this molecule would be represented by a system containing 4 and 5 particles maximum. And some of the preliminary results show how, from this top view of palmitic acid, how this system start to form islands, and they spontaneously can form these islands, and then it's a very promising tool to tackle this system. We need to check first that the potential works for all the systems that I show here, and extend the calculations to mixtures with different compositions of monomers. So far, they reproduce quite well the sulfur density coverage that is observed in the atomistic simulations. So, conclusion from this part of the talk is that the hydroxyl groups play an important role in the interaction to grow the biopolymer. The cell assembly process of UNICAMP provides an adequate arrangement to ease the spontaneous esterification in mild conditions. And finally, the aleuritic acid is a promising monomer for the fabrication of these putty biomimetic polyesters. So I will move on the second part of the presentation. This is an interesting current work motivated by experimental results produced by Professor Elson Longo. And we can see here in this first paper about the topic, in which this structure, silver tungsten crystal, when they are observed by different microscopic techniques, they develop different structures. So it would be initially the tungsten, the silver tungsten, and after time, some nanorods, nanoparticle, nanostructure are formed. What is the origin of this structure? This is again another silver tungsten, and after a time, we see this bit. That when we analyze the composition, we find that mainly is made of silver, metallic silver. So when we just observe, we are injecting electron. We are irradiating electron on the sample, and then this originates a process in which silver, metallic silver, is grown. This is a very novel system. Again, here we can see these very nice crystals, and they are irradiated by electrons, and then along time some nanoparticles are formed, even nanorods. So the amount of silver is quite huge, as I will show later. Let's start just looking at the structure of the bulk silver tungsten. This is a metal oxide a bit tricky in the sense that it's made of a distorted polyhedra. So the structure is quite complex. We can find only one distorted polyhedra for tungstate, but for silver we can find 4 different coordination numbers, and even for this polyhedra, different configurations of polyhedra. So the system is quite interesting. So we perform DFT calculation using crystal and using VASP to reproduce, first of all, the electronic structure, the band gap of this system, and also to reproduce this kind of polyhedron. Let's look at the most stable surface at the system, which is the 100 surface. These are 2 different models with different thick. We have been trying to testing if this smaller one, which is more cheaper computationally speaking, if it's comparable, all the results between both. And yes, we can use this for the topic I'm talking about now. We find at the surface different silver atoms with different also polyhedra or coordination numbers. It's possible to how can understand the nucleation process in this system when we just inject electron from the microscope. First of all, we try to see if this silver atom can exit the surface. If they can just diffuse outward and initiating this process. To do this, the first thing is find a stable position out of the surface. And we find that all of the silver atoms behave in the same way. There are some silver atoms which are very difficult to get out. They are very stable and they don't, if we try to move them outside, they get back very quickly in the relaxation optimization process. But we find some of the silver atoms that can diffuse outside. This is interesting because of the observation, experimental observation. If we expect that all the silver atoms can diffuse outside, what we would expect in the experiment is just an amorphization process of the substrate. But we don't find this. Experimentally, we find the formation of large metallic silver structures, and the substrate still keeps a silver tungstate structure, very crystalline. So this result is interesting because shows that not all of the silver atoms can initiate the process. We found these 2 silver atoms, and we performed Nagel elastic band calculation to estimate precisely the energy barrier for these atoms to diffuse outside. And we find that the barrier is very low. Actually, it's around 0.1 electron volt, which is relatively low. But this is, well, I didn't show for the other silver atom, but the barrier is similar to this. But what happens when we also facilitate the process injecting electrons from the microscope? We have performed a simulation just including this electron effect, and we find that the diffusion path has no barrier, no energy barrier. So we find that the silver atom just will follow this reaction coordinate with no energy cost. So if we compare both situations, the small barrier can be even reduced by the effect of the electron in the system. Another surface, which is also is not the most stable, but is very frequent in the morphology of the metal oxide, is the 001. After cleaving the bulk, we find this morphology for this surface, in which we again find different clusters with different coordination numbers. This is a rotative view of this layer, showing the cluster which are here separately, just to have a picture of how complex the system becomes. But even this is the surface relaxed after cleaving. However, I am using ab initio molecular dynamic simulations. We found that this surface can reconstruct the atoms from the first layer and rearrange strongly, and they can change to this kind of clusters now. So we again try to study the surface, looking at the different silver atoms that can initiate the diffusion process. And we found again some atoms that can diffuse easily and others that remain in the pole positions. And we found that the energy barrier is very low too. And even we find this minimum for this case, or for this situation, we again, the energy barrier is lower than 0.06 electron volt, which is quite low. And we find this minimum again. This can be related to another experimental aspect, that is that in this system sometimes the nanostructure that grows, sometimes they can get back. It's not always the same process. It's very rich. Phenomenologically, the situations are quite diverse. But we find some situation like this, that some structure can emerge, and after a time they get back. This could be a reversible process, pathway to explain this. Another observation in this nanostructure, silver nanostructure, in this nanorod, if we just look in detail with transmission electron microscopy, we find this. What is this? This is well, this is a silver nanorod growth, but this corresponds to silver clusters. So the electron irradiation also generate a silver cluster around the nanorods, and this corresponds to different images from a movie that we'll see. This movie is one example of this structure with the arrow close to the nanorod, just focusing on different nanoclusters. These nanoclusters can behave when irradiated by electron differently. Sometimes the cluster can get back and join the nanorod. Sometimes they can sinter, like this sample. Sometimes they get close, but they repel and go back. And there are different possibilities. In this case, it's clear how the clusters get close and join. We're trying to analyze the reason why sometimes they collide and they sinterize, why they don't. Probably it's related to the different morphology of the crystal. So we have been recently starting doing simulations with different nanoclusters and simulating the collision between different sizes. Try to reproduce some of the situations in the experiment to understand how the atomistic structure of the nanocluster is, and find if there are favorable phases that allow the interactions or not. So, just to end the talk, some of the application of these materials, the first one is the one where the system was explored initially. So, when in the group, the specialists in photoluminescent materials were trying to understand this material, and then they observed the appearance of this phenomenon. So this is a photoluminescent material, but after the silver bronze also can act as an antibacterial agent. So it has a very interesting behavior in this sense because of the silver filament. And also another application is like as an ozone gas sensor. It behaves, it has a very nice behavior, even detecting very low concentrations of ozone. So, just two conclusions from this part. The first is how the electron irradiation can generate the formation of silver nanostructure on silver tungsten, and how the DFT calculation can help to understand the initial process of the diffusion of surface atoms to outward and without energy barrier, and how the electron irradiation also accelerates the process, reducing the energy barrier. Some concluding remarks are how classical and quantum approaches can help to experimentally to have a more atomistic level and understand some experimental result, devising new experiments, replacing unavailable experiments, and finally to get new predictions. So just some the collaborators or the first part of the talk, people from Spain, Instituto do CSIC, do Consejo Superior de Investigaciones Científicas. For the second part of the talk, people from UNICAMP, for Physics Institute, and also theoretician people that support also the theoretical part from the Universidad Jaume I in Spain as well, and people, the experimental people from the Instituto Nacional de Ciencias de los Materiales y Nanotecnología. Funding mainly for the cutting part from the ministry government in Spain and the Junta de Andalucía in Spain as well, and from the second part from FAPESPI mainly, and some support from the CEPID too. And thank you very much for
We are open for questions now. For the nanoparticle experiments, at what voltage low voltage, high voltage?
I don't remember exactly all the voltage, but they perform also in studying with the looking at the dependence with the voltage. So for a very broad range of voltages, and then the experiment is independent, the result on the voltage.
But do you know that at high voltage, if the electron beam has enough kinetic energy to sputter the sample or not?
No.
It's below,
it's
below the pressure.
So, in the first part of the experiments, in the one that I think is the Q10, where there's a carboxylate group and there's an OH group, you mentioned, but that seems a pretty good candidate for either QMMM calculations or reactions. Yeah? So that you can investigate the mechanism of the reaction. Is the mechanism no?
No, it's totally novel in the sense that there are no in the literature people from computational side trying to simulate this sort of system. We found in another area similar things using reactive force fields. Reactive force field for another kind of polymers, trying to look at the reaction, but not for this specific system.
Then I have one question. It seems to me that the reactive force fields describes well the sort of things that Bill was talked about, or other. We have energetics, high energetics, but you have some energy coming from somewhere, then that the reaction will proceed. But I'm not sure if that is a substitute for an actual quantum calculation, where the energy comes from the thermal fluctuations. Maybe you that have more experience with that sort of problems.
Yeah, I agree. I never tried to use reactive force fields. I don't know how they behave. I'm not sure. Maybe it's an alternative to look at this. We were thinking about using a quantum approach for this. Maybe using quantum QMMM. That's a possibility that we are trying to use eventually, because I think it would be very easy with this, at least to perform the reactions.
May I comment to this? I disagree. In fact, the reaction was developed to handle hydrocarbon reaction, so it works pretty well for the condition you mentioned.
It does.
Yeah.
So even without lots of thermal fluctuations, thermal energy, this is.
We did like the hydrogenation of graphene, and we don't use that much.
We don't use it.
Yeah. Oxidation is quite hard for reactant, but we can handle.
And the problem with reacts, it was supposed to be universal, but people now are in a hurry, so you have many subsets of parameters. And if you use the same method with different parameters, you got different results, which is bad. So, but this is because people allow it. In principle, after all the parameterization, it should behave like the universal for classical molecular diagram. So, in this sense, you need to be careful. In principle, it's a universal process. You can calibrate with DFT, you can calibrate with experimental data, and it should work very well for even explosive nowadays. The yield is going through an explosion with reacts.
Well, once you have lots of energy to put into the system.
But let's go to the real test. If you do simulation for a small molecule hydrocarbon kind, the error bar in the heat of formation from the simulations are in the same range, about 10, 15 kilocalories or more than the experimental. So it's pretty impressive. The small molecule. Clearly should be using more, because then you start gaining more data. Perhaps
Maybe you need some way to accelerate the reaction, but
Right,
just time.
This is a very nice testing example to try different alternatives, because it's actually also the classical atomistic is very close to the reaction. It gets you just at the end. So it's just only.
yeah, but if you're looking for how the part will change the radiation or the chemical reaction, you cannot do it with DFT. It's too expensive.
Yeah. No, DFT definitely will be an affordable, but UNM actually.
Let's